My name is Ed Lipkin. I'm in the Department of Medicine here at the University, Division of Metabolism, Endocrinology, and Nutrition, based predominantly at University of Washington Medical Center. And for the next 50 minutes, what we are going to focus on is the potential use and misuse of laboratory tests for assessment of nutritional status. The format, I usually am a little bit more informal, but I think we should try and save questions for the end. I'll try and limit it to about 50 minutes, and then we'll have about 10 minutes for any questions and discussions. As far as disclosures, very straightforward. I have no financial conflicts of interest, nor do I get any kickback from laboratory medicine for things I might say in this presentation, okay? But like a lot of potential challenges and opportunities, there actually was a single most important event that kind of piqued my interest in trying to do this for laboratory medicine. So there was a seminal event. It was specifically, and I don't mean to put him on the spot here, but it was an email from Dr. Mike Astian in Lab Medicine. What he was inquiring about was the utility of a variety of nutritional panels offered by a specific company, illustrated on the slide, but it is not intended, this is a generic talk, okay? There are many such competitors to this company out there in the private realm at this point. And he asked me very specifically to review some online tests which were offered through this company. The three tests he wished me to take a look at was a component called Nutrition Eval. Okay, that's number one. He also wanted me to look at antioxidants with coenzyme Q10, and another one entitled Oxidative Stress Analysis. And I thought, oh, those are very interesting topics. And then he suggested a website that I consult in order to really get the information directly from the company, which was quite full of all sorts of interesting information. So I specifically focused first on nutrition evaluation, and what I was able to identify is that as part of this panel of laboratory tests, they offered to analyze 150 metabolites as part of the one panel, consisting of compounds, metabolites, and elements of interest. And then there also is a portion of the web page which generated a rather large analysis, 16 pages total printed, of 12-point type, okay? And of this 16 pages of output, there were 2 pages that basically made diagnoses. In other words, it suggested specific vitamin and mineral supplement schedule to meet deficiencies identified in this ghost patient, okay? And it also provided 10 pages of analysis of the results which substantiated their recommendations in terms of vitamin and mineral supplements. And I thought, whoa, this was really a topic worth taking on. Another precipitating event. I don't know how many of you actually have had the opportunity to work with this figure, but Bob LeBay also is an important contributor to my motivation to do this. I've known Bob for a large number of years. He's an incredibly modest individual, but he actually was former head of laboratory medicine here at the U in an era when it was probably limited to 2 rooms in the basement, I would imagine. He not only was seminal in developing laboratory medicine at this institution, primarily through Harborview initially, but what many people don't appreciate is how much of a truly innovative pioneer he was in the application of biochemical measures to nutrition assessment. So he is very modest about it, but his contribution has been substantial. And he is a primary motivator behind me wanting to try to talk to you about this issue. I'd like first to take the time out and review with you, just from a historical perspective, what has been the evolution of our ability to assess nutritional assessment. I would date its primary onset approximately to the early 1900s. And the focus at that time, as you may be aware, was the assessment of energy expenditure as a major interest of the armed forces. These resulted in the seminal work of people such as Harrison Benedict, which you may be acquainted with. Then we can fast forward to another major conflict, World War II in the 1950s, Korea, and actually the focus shifted to concerns for optimizing refeeding of malnourished war survivors. But of course, you had to identify those individuals who were good candidates for refeeding in the first place. In the 1960s to 80s, there was a development of anthropometric and laboratory measures adaptable to the field. The most obvious example is a simple spun hematocrit, screening for anemia in third world populations. It was applicable to large-scale screenings in third world populations and widely applied. Then we could move forward to the N. Haynes effort within the United States to define the nutrient status of the population at large. There's a variety of other health measures. And then the 1980s, application of these techniques to hospitalized patients, bringing it back home to the United States. Critically ill populations and specialized populations requiring nutrition support were often the object of investigations at this time. And now the current era, in fact, has moved well beyond these historical roots and has evolved into the development of platform or so-called multiplex type platforms for assays of multiple metabolites all at the same time. The application of multivariate regression equations to nutrition assessment, and in the current era, most importantly, the evolution of genomics, proteomics, now metabolomics, and there's even a brand new. Has anybody heard of microbolomics? Okay, so this is characterizing the actually content of your gut microflora to your metabolome. So there's yet another omic on the horizon. And this is very much an evolution and direction that things are currently going that is current. You may need to realize when you look at this from a historical perspective that much of the older literature from which I will share with you some results was confounded by the high prevalence of infection in malnourished populations and its impact on measures of nutritional status, which is huge. The traditional approach to assessment of nutritional status really relied on these three specific aspects. One was some way to measure stores of essential nutrients. Number two was trying to advance our ability to quantify intakes of nutrients. And third was somehow to get a handle on quantifying the net losses of nutrients from the body. All three of these approaches were utilized. How did laboratory medicine actually contribute to this sort of approach? Well, substantially. If you just focus on saying what somebody's body composition was, in other words, trying to quantify their fat and lean tissue losses or their components of their total body mass, and in fact, measurement of serum and urine creatinine is a direct reflection of muscle mass, has been well validated in innumerable older studies. Also, if you look at the assessment of body stores, advances contributed by lab medicine, including the ability to measure iron and copper content of liver biopsy, significantly advancing the diagnosis of hemochromatosis and Wilson's disease. Some of that actually done here. The development of an assay such as a seroferritin measure, which has been validated as a very good surrogate measure of iron stores in peripheral stores. Serum albumin is a measure of visceral protein mass. And if you look at the whole issue of trying to assess and quantify dietary intakes, well, there are measures that have been around for a long time, but actually are quite helpful. A very straightforward blood urea nitrogen, measurements of transthyretin, urine urea nitrogen, total urinary nitrogen, all reflect to a certain degree protein intakes in a person who is stable. And if you look at the issue of trying to quantify expenditure, there are also contributions of laboratory medicine because measurement of urine urea nitrogen or total urinary nitrogen reflects the extent of protein breakdown in the whole body. And then independent of lab medicine, of course, calorimetry has been applied in various forms as a measure of total energy expenditure. There's lots of older lab tests used in nutritional assessment. All of them certainly are available on a widespread basis currently. They're listed on this slide. It's simply a potpourri assortment of different laboratory tests that certainly can be utilized in the current era. They consist of a variety of serum measures, measures from blood counts, and some other specialized tests such as measurement of vitamin levels, trace elements, and activity coefficients for vitamins such as thiamine and pyridoxine. They all have a certain degree of applicability. I'd like to focus just on one specific example first. That is measurement of the serum albumin and what it may tell you. This happens to be a single patient from Harborview Surgical ICU, and it illustrates that albumin, one, has some utility, but its interpretation is quite confounded, and yet the literature about this historically has been enormous. This illustrates an individual who was admitted there with an infection. The infection is day one. Diagnosis of infection. You see the response of what happened to the serum albumin as a function of time in this particular patient. Also characteristic of infection, however, and also well validated by the serum marker of C-reactive protein, is there is a robust inflammatory response to the infection that more or less parallels the plummeting of the serum albumin level. All of this was advanced through monitoring by laboratory medicine. This patient was treated in a definitive fashion with both surgery and antibiotics, and you might wonder, well, gee, if this patient was behind the 8 ball because of her albumin falling to low levels, wouldn't it respond to being cured of her underlying problem? And the irony is that although C-reactive proteins fall rather robustly in response to definitive treatment, in fact, there is almost no change in the serum albumin for a prolonged period of time after resolution of the infectious process. So although albumin has some utility as a marker of overall health and nutrient intake, its response to cure is very, very sluggish and slow and inadequate. On the other hand, even though the C-reactive albumin level might be falling, the C-reactive protein might be falling as the patient improves, and albumin levels almost never change. Another assay developed here, transthyretin, or measurement of prealbumin, in fact shows a very robust and significant response to adequate feeding and resolution of the patient's underlying disease. So another example where monitoring through the lab actually helped track what was evolving in this patient from a clinical point of view, and can be of some help in putting together the big picture illustrated here all together of what happens to all these markers of the different phases of disease utilizing laboratory measures. Another classic example of where measurement of a simple albumin is of some help is in the diagnosis of kwashiorkor. Again, this is a post-surgical patient. She has classic findings of what would be called adult kwashiorkor, and yet absolutely characteristic as being able to document hypoalbuminemia as part and parcel of this lady's clinical presentation. It's hard to make the diagnosis without having access to at least one measure of the serum albumin. The other factor that we know relatively well from the serum albumin level is that although it is an insufficient marker of poor nutritional status in and of itself, completely out of clinical context, it is a relatively reliable marker of who's going to die during a period of hospitalization. This is one of innumerable historical series which documented a very clear and inverse relationship between the serum albumin level and prospective mortality rates in hospitalized individual, and well validated in a variety of other clinical situations. Besides these kinds of static measures, though, there have been applied a variety of more functional lab tests, is the term I would like to apply, in nutritional assessment. Why functional? Because they actually do measure some concrete physiological or biochemical process. Classic example would be prothrombin time. But also, again, developed here at the university would be the application of a test such as the zinc protoporphyrin to heme ratio. And then there, of course, are activity coefficients which are useful in the assessment of vitamin status in some selected measures. Despite those types of laboratory-based functional measures, there's a whole variety that have been applied to nutrition assessment, though, that could be said to be of equal importance. That is some objective measure of muscle strength. And the gold standards have been such things as hand grip strength, respiratory mechanics. Specifically, you can translate that into the work or energy expenditure of breathing, and also various forms of muscle stimulation. Specifically, one that's been utilized is this little thing here for research purposes. And then there are other functional measures such as of immune status or skin testing, where you're simply looking for anergy in response to planting skin test antigens on individuals. But again, another very functional measure. There are specific advances that have been spearheaded here at the University of Washington in nutritional biochemistry. Zinc protoporphyrin to heme ratio and its utility in the assessment as a marker of anemia has been largely developed here and applied somewhat even worldwide for screening purposes. But it is a superb marker of the efficiency of iron utilization. Another test that was put online and developed here specifically is the ability to take biopsy specimens and actually get meaningful iron content from tissue biopsies, whole tissue. Another test that was developed specifically here is the measurement of urinary isoprostanes. As it turns out, if you're looking for a more specific and potentially sensitive marker of lipid oxidation, this is one of the better ones around. Another measure, at least at the university, that has been utilized is actually quantification of red blood cell fatty acids, which is not a bad biomarker of fatty acid intake in your diet. And lastly, largely at the Fred Hutch, but also here, there has been the development of various markers of measuring serum carotenoids by HPLC, utilized effectively as biomarkers of vegetable intake. Specifically, there are phytochemicals in vegetables such as lutein, zeaxanthine, beta-cyptoxanthine, beta-carotene, and lycopene. I actually got all of those out without stumbling too much, are all good biomarkers of your vegetable intake. As far as where this type of assessment has gone over the years, though, rather than relying on single measures, there have been attempts to take these multiple measures and actually develop multivariable prediction equations to diagnose either impending mortality or impaired nutritional status. So they have taken single variables and tried to distill them into multivariable equations. A typical example, two typical examples of multivariable regressions are just illustrated here for discussion purposes. This is one. They both happen to be from surgical series. You'll notice that albumin is a major part of this equation. It also involves a measurement of transferrin in the blood, combined with measurements of triceps skin fold, a measure of subcutaneous fat, and delayed hypersensitivity. Second measure you'll also note also contains albumin as one of the regression variables. Again contains delayed hypersensitivity, sepsis, and diagnosis. These types of investigators were always able to show that these kind of multivariable equations did a better job than just using a simple measure like albumin in order to predict either mortality or malnutrition. So they seem to have some value. Only one of these two, though, actually reported its positive predictive value, which was not bad, 72% for mortality. But it's a segue, a lead-in to the next slide, because there are lots of limitations to this kind of data in the literature. The regression equations that one can find and are published out there actually have few data on its true positive predictive value. A lot of the groundwork was just not done that you could actually talk about that in any meaningful fashion. And there also is little data on receiver-operator characteristics of equations. And I'd like to review with you a little bit what exactly that means and how it is, one, lacking in many nutritional measures, but how it maybe should be added to the things that need to be assessed before really believing about tests that you're putting on lines. As far as what is a receiver-operator characteristic, this is well described in the literature. It largely comes from trying to do research on biomarkers for tumor identification and tumor growth. This shows you an example from pancreatic cancer, and the data is actually the antigen CA19-9, which is a marker for pancreatic cancer. The histogram at the top illustrates the data set for a large population of screened individuals simply by those that had surgical confirmation of disease, illustrated in the dark color, okay? and the individuals who in fact were determined not to have pancreatic cancer. Then they selected various cutoff points for what separates these two populations along the x-axis. And utilizing that plus the raw data, they are able to calculate both the true positive rate for diagnosing pancreatic cancer as well as the potentially false positive rate for diagnosing pancreatic cancer and illustrate it in a graphical fashion at the bottom here, utilizing the raw data. What is characteristic of a test that is absolutely no good, in other words, it doesn't discriminate between disease or no disease, is a straight line through the center. If in fact this test behaves very well in discriminating between disease or not having disease, you get what is called a positive curve. It has a shape like this, and a very finite area under the curve in comparison to the straight line. That quantity actually is a good measure of how good this test performs in real life in separating pancreatic cancer from non-pancreatic cancer. Yet this type of thing is oftentimes completely lacking from these types of measures, of biochemical measures. So what I would say is that biostatistical principles learned from tumor markers have a lot to contribute to this whole area, but actually it tends to be completely lacking or deficient when one looks at the literature about measures of nutritional status. Because the ideal measurement that you want to develop and put online is both sensitive and specific. It needs to be both, actually, if it's going to be of great utility. In that usually there's an inverse relationship between sensitivity and specificity. Or very specifically, as sensitivity is increased, specificity is reduced. More associations are identified that may have no physiological impact whatsoever. So there is a balance here, and it needs to be defined. Also, maintaining high specificity or maintaining the ability to detect low false positive rate is a high priority for population screening, but may lead to underdiagnosing conditions which benefit from early intervention. So characterizing this type of property of any measure is an important clinical issue. As far as the application of statistical principles to nutritional markers, well, as I alluded to, older studies use nothing other than simple association, linear correlations, was there a relationship or not. Better studies, if you take the paradigm of developing biomarkers from tumor presence and growth, a lot of the seminal work having been done by a biostatistician here, Margaret Pepe, in the early 2000s, developed a variety of measures that ought to be applied to the statistical veracity of a tumor marker. And that was the ability to define sensitivity, specificity, all is defined on the slide. The sensitivity is the true positive rate. The equation for calculating is also listed on the slide. Specificity is the number of true positives, but also a false positive rate and being able to characterize the receiver-operator characteristics. She actually elaborated 5 phases of tumor marker development, which she felt were important if one was to believe the veracity of the findings from these tumor markers. And they all have a fair degree of support, at least from the tumor literature, about their importance in being able to say the utility of the tests. They're outlined on the slide here. Specifically, they rely on, number one, ascertaining how well any specific measure distinguishes between cases and controls. How well does your biomarker distinguish between these two situations? Ideally, measures are binary. Neither you're either plus or you're minus. You're either malnourished or you're normally nourished. There is a confounding issue here, though, of course, as we all know that most biological measures, however, are not binary whatsoever. They are continuous variables. A classic example as a physiological measure would be blood pressure. We used to think there is good blood pressure, there is bad blood pressure. There is no such thing. We are all different, and it is a continuous spectrum. And in fact, people who have almost no blood pressure probably live the longest. So there are lots of examples where biological variables are not binary. Their true positive rate is the proportion of case subjects who are biomarker positive. That's the ideal, that it should be as high as possible. False positive rate is the proportion of control subjects who are biomarker positive. And obviously, ideally, you would like none of those. You don't want any of those to be picked up by your test. And of course, receiver-operator characteristic curves are a plot of actually true positive rate versus false positive rate. What is very interesting about this statistics when reporting the veracity of a biochemical measure is it has nothing to do with the scale of the test. So it doesn't matter whether you're working on a nanomolar quantity or you're working on a gram quantity in terms of the measure itself. It is somewhat measurement independent. A rare thing, actually, for this kind of variable. There are lots of problems in trying to develop these kinds of markers related to nutritional status, and I'll take again another analogy. The analogy would be, imagine looking at gene expression profile, which I think we've all tried to do in various forms in our professional careers, based on microarrays in yielding information regarding expression of thousands of genes. It literally looks like a light show. If there is even a small amount of noise concerning the level of expression for any one given gene, then imagine the amount of noise generated in the entire expression profile. So this is just one example of one technology and how it may or may not be a difficult issue in trying to make sense out of associations with nutritional status. Variation is a direct reflection of its stability over time and reproducibility also, which may be a confounding factor. And the other factor, which we oftentimes forget, is we report the results, but actually descriptions of what is the control, what are you comparing it to, sometimes is not as well done as it needs to be. Well, where has all this evolved in the current era? There is a movement, and it was reflected somewhat by the initial email I shared with you that reflected like, gee, somebody in that lab is trying to develop the equivalent of what we call currently metabolomics, or a top-down approach to measuring nutritional status utilizing determinations of a large number of metabolites. And is this even feasible or possible in the current era? And in fact, there actually are examples of how this kind of approach has been applied successfully to nutrition. But we are talking about a field in its very infant stages of development, and I have to tell you, my honest professional opinion is it is not ready for prime time. But I will tell you what its current status is. There are essential nutrients for which applying this type of top-down approach actually has been very fruitful. I'll share with you one specific example from a paper published regarding celiac disease. Okay, this is a degenerative disease of the gastrointestinal lining, and there is one very interesting study out there about the application of metabolomics in celiac disease. It has been very fruitful in the application of examining the status of vitamin D nutriture. So as a biomarker of supplement use or your vitamin D stores, actually this type of technology has been applied very successfully to that type of measure. And there are some metabolic studies that have been able to characterize very distinct phenotypes that differ in terms of their metabolism. So I will share with you some specific studies that have focused largely on the intestine as an organ, but also adipose tissue, fat tissue, and also skeletal muscle, applying this type of technology. Well, first of all, a definition. What is metabolomics? I always have to go back to Google in order to figure out what people are talking about sometimes. So this was downloaded from the web. The global analysis of metabolites, small molecules generated in the process of metabolism that represent the sum total of all metabolic pathways in an organism. That is quite a tall order. With a focus on the identification of each pathway and its role in the organism's function. And this is from 2009, so you're not talking about a long-standing history here, okay? What you need to be aware of, however, is that as far as the NIH is concerned, this is really important technology to advance, to do research on and is supported by a huge investment of federal money at this point in time. It's been incorporated into the NIH roadmap as a, quote, core technology in the overall initiative to guide the development of and assist in delivering solutions to human metabolic disease. Now, is that a tall order? So this is supposed to be the technology that will advance our ability to cure cancer, prevent type 2 diabetes, and cure coronary artery disease, okay, among just a few. As far as what is the metabolome, well, then it gets a little bit mushy. I shared with you the definition, but the reality is it is not a physical structure. The beautiful thing about genomics, trying to define the genetic structure of a particular phenotype, is it's a concrete entity. It's a series of base pairs. All you have to do is characterize what the physical structure is, and that is the knowledge. The knowledge is in the structure. And yet here we're talking about something, and I'm telling you there is no structure. Okay, we are the ones who actually have to create the structure. But doesn't that make you a little nervous? All right, it's a finite number of basic molecules of human metabolism that are present in relatively constant amounts. So that's an important property that you would use to select such a candidate, without which life is not possible. So there it almost fits the rubric of an essential nutrient. You take it away from somebody, they're dead. Very straightforward. And are characteristic of a specific phenotype. Now there's the big challenge. So we do all of the former two parts of this. In other words, it's present in relatively constant amounts. We're able to show that in fact life is not possible without it. But can you really associate it with some specific phenotype that's either a metabolic pathway or a disease process like type 2 diabetes? And that is really the rub. Also, it is maintained in homeostasis, so there's regulation. Regulation has to be characteristic of the metabolome, wherein variations in levels are small in comparative to its relative abundance. In other words, it might be present in a large quantity as an entity, if you can define it, but actually its variation is very small in relationship to its net amount. Well, what are the components of metabolomics and how has this evolved? In fact, what they are trying to do is meld many different technologies into developing this concept of what are metabolomes. And they are all illustrated on this slide. So that they have taken data from a variety of technical advances, laboratory-wise, metabolomics, proteomics, transcriptomics, which if you want to distill it down to something concrete, we're talking about RNA and RNA species here, genomics and various omics, but then with a variety of other functional measures, all in the upper tier here, and developing a metabolic phenotype which has interactions across a wide variety of intrinsic issues, including the genotype, lifestyle, other factors, activity level, gut microflora, to create the total genome. It requires a massive ability for data storage and transformation of the data in order to construct what would be thought of as the essential metabolome. There are a variety of factors which impact this, which make the challenge daunting. So in the center is our ability to construct what would be called a metabolome in the center of the slide. But the intrinsic factors that would influence this are many, including body composition, tissue turnover, resting metabolic rate, age, the genotype of the individual. In other words, not just what your endogenous genotype is, but perhaps how it affects nutrient absorption, utilization, and also losses in the body. Also, health status of the individual, reproductive status, and diurnal cycle. There are also a variety of extrinsic factors, perhaps, that we would like to know more about as the object of being able to define the metabolome, such as diet, drugs and their metabolism, physical activity impact, colonic flora, and also stress per se. But all of these are important factors that may play into the metabolome. As far as what we currently understand, our knowledge base is relatively limited. We are beginning to understand more and more the genetic contributions to absorption, utilization, and distribution of nutrients, as well as their metabolism and loss. We know a fair amount about the dietary components of what affects blood levels of various things. And we are beginning to explore what is the contribution of actually the commensals that live in your own body, since we support a population of a few billion actually on our own. But it has an integral relationship to your health and well-being in what you circulate actually in your bloodstream. So there is some advance at the level of understanding the microbiome. Just to illustrate, though, the complexities of trying to figure out what would be the impact of food per se on any such measures, this tries to illustrate it in a very specific factor. So not only is there the nutritional content of foods, but there also are non-nutrient components to foods, and we all know about them. They are phytochemicals synthesized specifically by plants, okay? But there are also a variety of man-made components that contribute in our food intake. Some are intentionally added, some are contaminants that we're just beginning to understand. On the nutrient side per se, there's certainly that derived from the foods per se, but also our food supply is supplemented in a huge fashion. So what comes from supplementation versus the food supply itself? Just to tease out all of these different contributors is a daunting undertaking in and of itself. There are focuses of current efforts wherein there have been some significant advances. So this is advocated as a paradigm for how you might think of what a metabolome is. And it simply is, what is the difference between the fed and the fasting state? Okay, you would think that would be very straightforward, and actually we do understand a fair amount about it. So characteristic of the fasting state, if you focus on the left side of the slide, would be an empty stomach and intestine, so potentially imaging or functional studies of intestinal metabolism and function would be helpful in constructing this type of metabolome. Also characteristic, however, would be specific effects on the liver and the stomach. Hunger is certainly activated by important hormones. A classic example currently would be considered ghrelin, GH-relin. There tends to be also mobilization of specific energy metabolizing signals, specifically from different stores such as fat and consisting of fatty acids. If you then compared this type of metabolic profile to the fed state, there are obviously differences which you could detect either by focusing on imaging studies of the involved organs, by characterizing what is in the blood, or even using more current technologies such as mass spectroscopy to do so, and or measurements in urine. And it is done by comparing the fed state to the fasting state, which quite the opposite would involve filling of essential organs, a complete change in the profile of essential satiety promoting hormones, and essential metabolites mobilized from adipose tissue and or muscle. So this type of effort is possible and currently has been done. There are a variety of issues that confront you, though, if somebody calls you up and says, I want to measure X. First issue would be like, where? Where am I going to measure X? When you think about these kind of top-down approaches and sampling issues, there's a variety of different accesses to trying to measure all of these different metabolites, and they're all listed on this slide. They all have potential advantages or disadvantages, however. Of course, there's blood, urine, but also saliva, unfortunately poorly standardized. There is certainly access to feces, biopsy material under some circumstances, and also CSF fluid as a reflection of what's going on in the central nervous system. The biggest issue, though, is which compartment to sample. As I alluded to, they have pluses and minuses. The saliva is poorly standardized. Blood has a great advantage because it has a relatively high signal-to-noise ratio for metabolites of interest to non-metabolites from confounders such as plant phytochemicals and other chemicals arising from cooking. So blood has its advantages. Urine, on the other hand, has a relatively low signal-to-noise ratio. In other, the signal that you're really looking for that would be characteristic for your test. As an example of this, actually onion ingestion has been well studied. because onion contains metabolites called quercetin. It was characterized that actually 11 metabolites were unique to urine samples collected from individuals in a feeding study of onions, as opposed to 5 unique that were only identified in blood. So there are more such confounders in urine than there are in blood. Another major difference between blood and urine sampling is that, of course, lipid-soluble compounds, if that's what you're looking for, circulate only in the plasma and not in the urine. There's a variety of technological advances in lab medicine that are being applied to nutrition research, and certainly they are being utilized in this effort to try and develop metabolomes or further metabolomics. Multi-platform assays I've alluded to. There's been a tremendous advance in our capabilities with HPLC, but also NMR spectroscopy and mass spectroscopy, all applied to lab measures. These have been tremendous technical advances. An example of the application of NMR to analysis of metabolites in urine. This is just taking urine and being able to quantify the metabolites in a urine sample. And in fact, it shows you the entire profile of what they were able to identify from the straightforward NMR spectrum. This kind of boggled my mind when I read this article. I mean, the last time I did NMR, it was like an ultra-pure organic compound that had to be put in a special solvent as a single molecule in order to characterize its NMR spectrum. So the advances in spectroscopy with NMR have been rather tremendous. Identified here are 11 identifiable metabolites, all in urine, from one NMR spectrum. There are specific examples I'd like to show with you of the application of metabolomics applied to human disease phenotypes. The first one I already alluded to, celiac disease. So what is the technology that has been applied? It in fact was NMR identification of metabolites in urine and serum. And what they utilized was a case control comparison. Cases were compared to controls on and off a gluten-free diet, which you should be aware is able to induce a remission of celiac disease in gluten-sensitive individuals. And the result of this is that they were able to show in the serums of celiac disease patients, it was characterized by having lower levels of amino acids, lipids, pyruvate, and choline, and higher levels of glucose and ketone bodies. Whereas the urine of these subjects shows increased levels of very interesting, somewhat novel compounds, indoxyl sulfate, metahydroxyphenylpropionic acid, and phenylacetylglycine. What's interesting is those are all actually products of bacterial metabolism. Okay, but it was characteristic of the phenotype that defined celiac disease, at least in this case control study. Okay. Other examples, there has been an advance in trying to define what is the difference between exercise and non-exercise by applying metabolomics. The methods employed are somewhat interesting. It involved both gene and protein expression profiles in a mouse model. And the results of this were the ability to show that upregulation of AMP kinase activity or the administration of a pharmacological agent, PPAR gamma agonist, mimic the effect of exercise on gene expression and protein expression in the muscle per se. A kind of interesting comparison, but there were clear, definite differences between the phenotype of what was characteristic of exercise versus non-exercise muscle. Another example, focusing on a different tissue, was the ability to characterize the difference between basal and insulin-stimulated glucose muscle metabolism. This particular study utilized NMR quantification of ATP level in muscle biopsy specimens, and actually a mouse model of increased expression of peroxisome proliferator activated receptor Y cofactor, or PGC-1 alpha, and a dietary-induced insulin resistance. What they were able to show is actually a 2.4-fold increase in mitochondrial density with a 60% increase in ATPase synthesis, but reduced muscle insulin-stimulated glucose uptake. And we're very capable of distinguishing the two types of phenotypes by this type of approach. You may note that actually is somewhat of a paradoxical finding. but still quite interesting in this whole field. Other examples of omics that I wanted to share with you is applied to nutrition research. A very interesting study comparing the phenotype of mitochondrial overload and incomplete fatty acid oxidation, which is a very active area for what is controlling metabolism during acute phases of illness. This particular study utilized targeted metabolomics, so a slightly more focused approach than measuring everything under the sun. The techniques were gas chromatography, mass spectroscopy of fatty acids, in a fat-feeding model, rat model of insulin resistance, and also using a mouse myotube culture system in a mouse lacking malonyl-CoA decarboxylase, one of the rate-limiting enzymes in fatty acid synthesis. The result was the ability to show that on a high-fat diet, insulin resistance is prevented by restricting fatty acid uptake into muscle by these particular pathways. One last study I wanted to share with you is trying to distinguish between a phenotype of fat tissue deficient in fatty acid binding proteins versus normal fat tissue. So here the focus is on fat tissue. And this again used a targeted metabolomics approach, focusing specifically on lipids. In again a fatty acid binding protein deficient mouse model, the result was able to show that FABD deficient mice exhibited increased de novo lipogenesis, decreased dietary induced insulin resistance, and high levels of a circulating novel fatty acid, C16, 1, and 7 palmitoylate, a marker of increased fatty acid synthesis. These are just some specific examples of how this type of newer technology is being explored to try and define differences in metabolic phenotype. Okay? In summary, it's been a long road, but the current gold standard for assessment of nutritional status after all is said and done is relatively quaint because it still relies on a diet history, a physical examination, and focused measures of specific laboratory entities. But there are examples of disease phenotypes that seem to be facilitated by a shotgun characterization of metabolites, specifically celiac disease, and it clearly is the wave of the future. And in conclusion, there has been a tremendous advance in the application of newer technologies, specifically NMR, mass spectroscopy, and proteomics to the measurement of metabolites, elements, and key proteins. But the application of newer technologies to nutrition assessment, including metabolomics, is in its bare infancy. So that sounds daunting and a little bit overwhelming, but I also throw it to you a little bit as a fish because, boy, is there opportunity to advance this field. And I'd certainly like to open it up to questions and comments.
So I've got one question. You really sort of having highlighted some of the challenges and the problems in sort of defining what the metabolome is and trying to measure it. Is there data sort of linking metabolomics to genomics, which is sort of, I guess, even though genomics itself is as you alluded to, is at least a definable enterprise and a thing that can be pointed to and said, this is the genome. But are there good examples linking something which is metabolomics to genomic variants, for instance?
Well, there are actually a couple of members of the audience who probably know a little bit more even than I do about this, but of the examples I shared with you, the current status of that type of approach is you take a relatively defined genetic model, and then perhaps you apply some of these techniques that are applicable to metabolomics to define the metabolic profile. As far as like taking a broad population of just a specific phenotype and trying to integrate kind of a top-down genomics type of approach with this type of measurement of metabolites. I couldn't find any concrete examples to share with you where there's been actually a great deal of success in that vein. It's been a more structured kind of approach. You do a controlled intervention, changing the genome or developing a mouse model, and then you investigate the impact in terms of potential metabolite profiles. So that's my understanding of kind of where it stands currently. Would you disagree with that, Andy?
No, I think that's right on.
Yeah. So I asked Andy Hofnagel, one of my brilliant colleagues in lab medicine. Other questions? Yeah, Mark.
Yeah. Thank you for this broad overview. Is there progress being made, or are people approaching again, I guess, the question of sorting out nutritional status in the setting of chronic inflammation and sorting out nutrition from infection in terms of these, in terms of markers of nutrition per se that aren't affected by or can be somehow controlled for the effect of inflammation?
That's an excellent question. I don't know the honest answer to that in terms of the whole field at large. I'm more overwhelmed by some of the confounding issues when you start to examine really sick individuals. So as any of us who have ever dealt with a hospitalized patient, you do all these wonderful things and you think you've cured them, and maybe you could come up with some sort of case control comparison and make sense out of these types of comparisons. And then lo and behold, like the next day they have C. difficile from all the treatments you've thrown at them to cure their underlying infection. And maybe the C. diff has actually been there for 3 to 4 days, kind of perking along before it actually came to the fore as a clinical phenotype. So they are fraught with all sorts of other confounding issues related to treatments and changes in their endogenous bowel flora, all of which I could imagine would make any real comparison a daunting undertaking. How do you sort all of that out? At this point, all I can do is elaborate what I see as the confounding issues, and honestly, I don't have a clear vision out of that. I think if you could imagine some sort of disease process, and the people who focused on celiac disease, I actually thought did a brilliant thing in simply thinking that this was a reasonable model to investigate, because they could have a before and after intervention comparison to something that potentially was curative of the underlying phenotype. In other words, gluten restriction in the diet. So at least in that sort of situation, at least you have a reasonable intra-individual comparison group. It's before and after an intervention. There's a lot of other things missing from that kind of intervention, but the sort of thing you're advocating or trying to look at, I find very difficult. I guess if I could see any parallel about a way to approach that, it would be that you need to perhaps approach it from a defined genetic model that either changes various modulators of the inflammatory process and then perhaps perturb that somehow, but on top of that background to make a comparison. So maybe you would impose an acute infection on a background of a genetic variation in a mouse model. But I think that at least initially you would have to try and clearly separate those two issues instead of thinking that you could tackle both top-down to begin with.
So Ed, from
Yeah.
your perspective and in terms of nutritional status, what would be the most exciting lab test that we could offer you for the next couple of years? Where would you have us look to help you the most?
Well, I think you've already done some of them. So he gets a few kudos here. I think what this place did in terms of vitamin D determinations actually not only was an incredible piece of science, but it's actually become kind of the standard community-wide for what is a decent assay out there. And if you look at the history of what used to be the case, the bottom line is you could not believe 90% of what you were reading about vitamin D levels in older publications because it was a worthless test. So there's one specific example. If you really want to know kind of state-of-the-art application of some of these sort of things, if you read the publications of Jay Heineke, the ability of proteomics to define characteristic profiles of really unusual and quite interesting proteins that tend to be associated with a certain disease phenotype, in his particular instance, coronary artery disease being the phenotype, the advance has been incredible at elucidating all sorts of potentially interesting biomarkers that we had no idea might have some role in the overall process of coronary artery disease. So another example of something that already has been developed pretty incredibly at this institution would be the ability to do proteomics on specific entities such as HDL protein. And I could imagine that there may be, even in tumor biology, a whole slew of similarly analogous type situations where that type of approach could be very fruitful with those tumor markers. Sometimes there are things associated with these circulating particles that you wouldn't have had the slightest clue were actually lurking in your blood. And I found that to be incredibly exciting.
Any questions? Dr. Lipkin, thank you very much.
Thank you. Thank you very much.